Here we are in Panzironi's classroom explaining to us. And this episode, we are going to delve into a really very interesting topic. We are going to talk about insulin. Here it is, this hormone that is considered a lifesaver, but its excess is really a problem for our health. Right, Panzironi?
Yes, you are completely right, because insulin is a very powerful hormone that really saves our lives, but it also goes to alter a whole series of other hormones that it, in short, interacts with. So it's important to know and understand what it's all about, especially to unravel all those misguided dogmas of modern medicine.
And so let's get right into learning more about insulin with this video.
Insulin is a hormone produced by the beta cells of the islets of Langerhans within the pancreas, based on blood concentrations of glucose. In fact, glucose enters these cells mediated by a specific transporter, GLUT2. Within beta cells, the increase in ATP produced by glycolysis induces the closure of potassium channels and the increase in calcium and the consequent release of insulin by the Golgi apparatus. Insulin poured into the bloodstream binds to cell membrane receptors, promoting the distribution of GLUT4 across the membrane. GLUT4 are present in striated muscle fibrocells and adipocytes, cells defined as insulin-dependent. Insulin induces glycogen synthesis within muscle fibrous cells and promotes fatty acid synthesis in adipocyte cells. Insulin also has an anabolic action by promoting the production of the hormone IGF-1.
The protein subductors of the gates that are called GLUT2, depending on the amount of blood sugar in our blood, allow the production of insulin. Then the insulin, through other special GLUT IV ports inside the muscle fibrous cells and adipocytes, allow this glucose to flow out inside these cells, and thereby reducing blood glucose.
Look, Panzironi, you have always told us. You have always taught us that the only cells that use sugars are brain neurons and erythrocytes. But do these cells also have GLUT4?
No, absolutely they don't. And you made a correct observation, because actually the only cells that use in an exclusive form carbohydrates, therefore sugars like neurons and like erythrocytes, actually do not need insulin to use precisely sugars. If anything, we find these GLUT IVs in adipocytes and muscle fibrous cells, because they are those cells that are able to store more carbohydrates, and this should make us understand, especially it should make the so-called official medicine understand that insulin is precisely an emergency hormone. That is, it should save our lives.
Look, Panzironi. You said it must save our lives. In fact, you say that.
Basal insulin level in the blood deviates from the range of 6 and 30 microunits per liter. In addition, one can assess the proper functioning of insulin by taking the insulin curve test after the administration of 75 grams of glucose. The reference values of the insulin curve are in the range of 10 and 200 microunits per liter within the 120 minutes after the meal, while an overshoot of these parameters certifies the presence of hyperinsulinemia. The excess of insulin depends on the loss of sensitivity of cell receptors toward this hormone, which the body attempts to compensate for by increasing the production of insulin by the pancreas. The loss of GLUT IV sensitivity to the hormone insulin occurs mainly in muscle fibrocells as a result of mitochondrial dysfunction and the accumulation of fatty acids in the cell cytosol. Excess insulin is capable of causing severe metabolic alterations by interacting with leptin, with thyroid hormones, in the activity of mTOR, in the inhibition of sirtuins, in the promotion of cell duplication, in the stimulation of IGF-I.
Then, Panzironi, the video explained to us what the ranges are, because we have normal insulinemia, or those others are in hyperinsulinemia. So he told us about those two ranges.
Yes, certainly having a problem of hyperinsulinemia is serious. But I also have to tell you that the most serious problem is that the ranges that provide for less recognition of this condition are absolutely too excessive, because to go from, for example, to a basal insulinemia of 3 and 30 is really too high a range. Also, because we demonstrated during our 5 months to rebirth that people who were following the correct observational after 3 months had a basal insulin amount of 5 microunits per liter.
Here about range in the video, it was basically said that the range of insulin goes from 30 up to 200 microunits per liter after a meal based on the Mediterranean diet.
Well, yes, here we really are at the madness, medical madness, I might add, because you can think that it's normal, in short, an insulemia that goes from 10, even 30 to 200, that we're talking about amounts considerably higher than what is our basal insulemia. I think official medicine has to ask these questions because we are talking about people's lives and health here.
And to make us understand this, you have prepared a little practical test just to make us better understand the difference we have with the excess insulin when we go to eat a meal from the Mediterranean diet. Not surprisingly, we have our Alessio here next to us and in front of 2 cylinders. The first one we have already filled, and it indicates the amount of insulin present in the blood of those who follow the, obviously, Life 120 lifestyle and diet, and it is the one already with the fluid in it.
Yes, we considered that a basal insulinemia at 5 can be assessed at 10 after a Rivital-style diet at 120 for the simple fact that we do not introduce those so-called insulin carbohydrates. So we don't activate an overproduction of insulin.
Now, however, let's ask our Alessio to pour into the second cylinder the amount of insulin considered normal by so-called official medicine. The protocol medicine, and which should be present in our blood in the 100 minutes, in the 120, minutes after our meal. He is pouring, and of course I tell you, it is a proportion, it is not the real amount. Let's say in proportion from what we have in the cylinder with the lowest liquid in proportion, the other will be what we will have in the 120 minutes after a Mediterranean diet meal.
Yes, we have to consider that a normal classic dimia aranea meal that is composed of a pasta dish may be a main course accompanied with potatoes, maybe a fruit. Well, we introduce in this meal that we are told is so good for us. Well, we introduce something like 200 grams of carbohydrates versus instead an amount of carbohydrates that are introduced normally in a diet, not rivaling the 120. We reach precisely only 30 grams for one meal.
In the meantime, our Alessio continues to fill, and already from this difference we see the comparison between the 2 feeds practically is really impressive, and he is continuing to fill.
Well, it's impressive, but unfortunately it's the truth, because we're talking about an expected amount. We always repeat in a person who doesn't have hyperinsulinemia. So we're talking about 20 times what the amount of insulin is expected instead in people who follow the 120, even 40 times what the basal insulin. In short, really an insane average.
I also want to point out that in your new book, Living 120, the research is you have really devoted a whole chapter to hyperinsulinemia-related diseases, a whole chapter.
Absolutely, because the excess insulin goes to damage a little bit all the metabolic functions of our body. Even we go to alter the sirtuins. So we go to accelerate our aging, we go to alter thyroid hormones, we induce fat mass, and therefore the problem of obesity will lead to diabetes. In short, a whole series of pathologies that, by the way, are recognized in the hyperinsulinemia, but we have to remember that insulin that does this damage. So introducing 20 times the amount of insulin of a normal step, normal predicted our biology, certainly will make us sick with all those pathologies that we know, unfortunately, well today.
Always precise, our Adriano Panzironi. So thanks to our Alessio, thanks to our Adriano Panzironi.